This is the first pathophysiology lecture. My name is Domokos, Gerő, and I'll give you a talk about inflammation from the viewpoint of pathophysiology. I'll talk a little bit about the definition of inflammation first, then the majority of the lecture will be about the pathogenesis of inflammation, how inflammation is initiated in our body, and what cells are involved in this whole process. Then we'll see how this one translates into the clinical practice. So what's the diagnostic use of all of this knowledge, how we can use the signs of inflammation, and how we can use it in the therapy. So what are the anti-inflammatory therapies currently in use. And finally, I will give a few very simple examples of inflammatory diseases. So let's start with the inflammation. So how can we tell whether something is good or bad or healthy or unhealthy? If we can see a tissue injury, that's definitely unhealthy. Or if we can see an infection, that's unhealthy. On the other hand, the normal cell turnover in our body is absolutely healthy. We do not see any harmful event in that one. So if we look at these two elbows, there is an obvious difference between the two of them. The right-sided one is red, swollen, and probably painful. And we can ask the patient whether he feels hot or whether he can straighten or bend the arm, and whether he used an ice pack or Tylenol already just to reduce the pain. So definitely when we just look at it, it's clearly an inflamed elbow. It's an inflammation. On the other hand, the other one is absolutely healthy. So the difference between the two of them is simply defined by these classical signs. And all of this is taking care of the innate immune system of our body. So the classical inflammatory signs include fever, sufferability or warmth, pain and discomfort, and redness, swelling, and dysfunction of the part of the body which is inflamed. And usually what we can see is that the treatment, an anti-inflammatory treatment or antibiotics in infections, antipyretics are helpful. So let's see how the inflammation is triggered in our body. When there is a tissue injury or infection, it'll be detected by our body by specific cells, and they will send a message to mobilize effector cells to the place, and then they will accumulate at the site of injury or infection, and these inflammatory cells, these effector cells, will take care of the problem, remove the injured parts of the body, remove the infectious agents, and the whole thing is resolved within a couple of days. So this is the so-called inflammation or acute inflammation which happens in our body, and this whole thing is taken care of by the innate immune system. This is our first line of defense. Now, it is a rapid reaction. It takes a few hours and lasts only for a couple of days normally. We usually see, though, that there is an adaptive immune system developing afterwards, which is much slower than the innate immune system. But if the innate immune system cannot take care of these infections or injuries, the adaptive immune system will be turned on, and as a second line of defense will take care of the infection. We will take a deeper look into the first part, into the inflammation, the first line of defense. Usually we make a discrimination that within the first 3 days, we can see most of the innate immune system's action. And afterwards, we can see the activation of adaptive immune system. So if someone has a simple infection and the fever is gone within 3 days, we don't need to worry. Sometimes when we can see that the fever is still there and the symptoms are getting worse, then we've got to think of the inefficient clearance of those agents. And then we can see that the adaptive immune system is turned on. So what is the general concept of inflammation and the immunity in this case? There are danger signals, inflammatory triggers, which was the start of this whole reaction. For example, the lipopolysaccharide of bacteria, they will be sensed by receptors, specific receptors of the innate immune system. These sensor cells will send messages via cytokines to the effector cells and other parts of the body to start up a vascular reaction and to mobilize the effector cells and attract them to the site of injury or infection. These are the neutrophils, as we do know, and usually this one will end up with the removal of the injured cells or killing of the microorganism and wound healing. The visible signs of inflammation occur only after the effector cells are mobilized or the vascular reaction happens. Nothing is visible from the first part normally. So what are these danger signals? We classify them as PAMs and DAMs. PAMs are derived from pathogens. They are certain molecules coming from pathogens, pathogenic bacteria, which can be sensed by our body. They are called pathogen-associated molecular patterns. So these are commonly present in many agents and can be sensed easily by a few receptors. Prototypical PAMs include, for example, the lipopolysaccharide, LPS, but also the lipoteichoic acid, LTA, or proteoglycans, double-stranded RNA molecules or single-stranded RNA molecules, which are not commonly seen in our body, or the microbial DNA, the unmethylated CpG DNA, or flagellates. On the other hand, if there is a tissue injury, we can sense similar signals coming from the cells, or damaged tissue or damaged cells will release certain molecules, which we call damage-associated molecular patterns. They will be sensed by the innate immune system, and they will start up the same inflammatory reaction. The prototypical DAM molecule is HMGB1, but the S100 proteins, heat chain proteins, ATP, or uric acid will be included in this group as well. And definitely our normal cell turnover will release a very tiny amount of these molecules. So we do need to have a slightly higher concentration of these released locally. That's the stimulus, which will be sensed by the innate immune cells. And what is even stranger, that actually our body cannot discriminate between the PAMs and DAMs. They do not react the same way. Either there is an exogenous or an endogenous signal coming from somewhere. It'll be sensed the same way and the same reaction will happen. So it's impossible to see what is causing the problem. So let's start with endotoxin, the prototypical danger signal. It was called endotoxin because in the beginning they believed that this one was an internal material in bacteria, which was released only after the cell lysis, as opposed to the exotoxins produced by certain bacteria, for example, the diphtheria exotoxin. Endotoxin comes from the gram-negative bacteria, which is one class of bacteria. They can be stained by the gram stain, and broadly, the bacteria can be classified into gram-positive or gram-negative bacteria. Gram-negative bacteria will have endotoxin actually in the cell membrane. It's a lipopolysaccharide molecule. It's a sugar, a chain attached to a lipid in the membrane. And this sugar in the molecule is slightly different in each of these bacteria. So it's specific for certain bacterial strains. These molecules can induce a toxic reaction or actually kill the mice very rapidly, so within 1 day or 2 days, depending on the concentrations. They are very toxic. That's the reason why they were called antitoxins. In reality, this one doesn't really mean a toxin, because later on it was discovered that actually the response to LPS is an inherited trait. Certain mice were recognized in the late 60s and early 70s, and they were resistant to antitoxin. So when they applied even 100 times higher concentration of LPS, they could easily survive that one. So they believed that this one is a specific reaction to the LPS, which our body generates. So the LPS will induce a reaction in our body. It will not be directly toxic to our cells, but it will rather induce a reaction which is toxic to the whole organism. And the endotoxin sensitivity can be transferred from the endotoxin sensitive mice to the endotoxin resistant mice. And this trait could be localized to a single locus, the so-called LPL locus on chromosome 4 in mice. It was a single trait, a single gene, and it was shown as a simple Mendelian inheritance in mice. So 50% of the offspring were resistant, 50% sensitive to endotoxin. So what is this specialty in this LPS locus? It took a very long time. So it was in 1998 when Bruce Beitler's group discovered TLR4, the true receptor of LPS. He took the mice which were endotoxin sensitive and endotoxin resistant, and he discovered earlier that macrophages coming from the endotoxin sensitive mice produced huge amounts of TNF-alpha, whereas the endotoxin resistant strain doesn't produce TNF-alpha. So he looked closely to the LPS locus, and he found that there were only 2 genes in that locus, the PAPA gene and the TLR4. And since PAPA was not expressed by macrophages at all, TLR was the only possible suspect. And it turned out that actually it was TLR4. TLR4 is the receptor of LPS, and this one suffers a point mutation, or it's completely missing from these LPS resistant mice. TLR4 acts as a receptor, and it was later on further shown by using TLR4 knockout mice. So you comparing the white-eyed mice and those mice which had a TLR4 gene selectively knocked down. So they compared the survival of mice and TNF production. Actually, the TLR4 knockout mice will not produce TNF-alpha at all, whereas the white-eyed mice will produce TNF in response to LPS. It's the same reaction if they use the TLR4 antibody. So if they block the TLR4 action, those will not produce TNF. And expectable, the TLR4 knockout mice, or those mice that received anti-TLR4 antibody, could survive lethal doses of LPS. Soon afterwards, they discovered that LPS doesn't really act on TLR4 only, but there is a secondary part of this receptor complex, which is called MD2. MD2 makes a complex with TLR4. So the MD2 knockout mice did the same thing. So when they were treated with LPS, nothing happened. They didn't produce TNF and they didn't die. They survived the LPS shock. So how does it work? Actually, it's a little more complex because LPS is first of all binds to the LBP, the LPS binding protein in the serum. This one will carry over these molecules to CD14 in the cell membrane of macrophages, and the CD14 will present the LPS to the TLR4-MD2 receptor complex, and LPS binds to the MD2 portion of the molecule, and this one will induce the assembly of a very complex signaling cascade, which is called a midosome. The MYD88 protein is the closest part of the TLR4 receptor complex in the intracellular part, but this one will be joined by other adapters in the inside of the cells, and that one will start up an inflammatory signal in the cells. So what is this inflammatory signal? As expectable, if MYD88 is missing from the mice, that one could protect them, and there will be no TNF production, and the mice will survive the LPS shock. And this one is the NF-kappa B pathway, which is activated by the midosome and will lead to inflammatory cytokine production, TNF, IL-1, and IL-6. Unfortunately, NF-kappa B cannot be knocked out the same way as the other molecules because that's embryonic lethal. Instead, they use the I-kappa B gene transfer, so the blocking protein, which makes a complex with NF-kappa B in the cytoplasm, was overexpressed to block the lethal effect of NF-kappa B activation. So this is how it looks like. So whenever we see an LPS molecule or LPS molecules in our body, that one will be carried to the TLR4 receptor complex, and the midosome will be activated, and this one will induce NF-kappa B translocation into the nucleus, which will drive the TNF, IL-1, and IL-6 transcription in the nucleus. TNF and IL-6 are exported as proteins out of the cells, but the interleukin-1 needs a further activation step, and that one requires a so-called inflammasome. For a long time, they believed that the inflammasome was simultaneously activated by the LPS receptor complex. Now we believe that inflammasomes are rather activated by extracellular other stimuli or intracellular stimuli, which will sense minor changes in the homeostasis of the cells. So for example, if extracellular ATP molecules are present or extracellular DNA molecules, that one can induce the assembly of inflammasome, or free mitochondrial DNA will be released into the cytoplasm, or the potassium level will change inside the cells, that one can induce an inflammasome activation. And if the inflammasome is activated, caspase-1 will be active, and caspase-1 will cleave the pro-IL-1 protein and will produce the mature IL-1, which is secreted into the plasma. So this kind of cell death form, when the inflammasome is also activated, is called pyroptosis. So the caspases are activated just like in apoptosis, this special form is discriminated by this process. So we could see that the NF-kappa B activation led to cytokine production. These cytokines will be very important because actually the TNF alpha, as was discovered earlier again by Bruce Beutler, is produced in response to LPS. If TNF is not produced, then the mice can survive the shock. Bruce Beutler received the Nobel Prize in 2011 for all his work regarding the innate immunity. And some of the discoveries I mentioned earlier are represent a great part of this work. So LPS was found, first of all, to induce TNF production, and then back in 1986, he and Kevin Tracy had the idea, why don't we try whether TNF alpha by itself can induce a lethal reaction? So instead of LPS, they used recombinant TNF alpha, and they injected the animals with TNF alpha, and this one induced very rapid death in the animals. Even though sometimes they use lower dosages, that one induced a similarly dangerous reaction, and in those survivors, they could see a very severe inflammation all over the body in various tissues. Though if they used a TNF blocking antibody, there was no lethality at all, so the mice could survive the TNF-induced shock. But the TNF gene is not localized to the LPS shock, so they already knew that this one was something completely different. TNF is produced very rapidly in mice, and also in humans, in response to LPS. So the TNF alpha will normally reach a peak in the plasma within 90 minutes, and then it disappears. So within a few hours, by the time when we can usually see all the symptoms, there is no TNF in the serum. TNF and IL-6 and IL-1 are termed early mediators or early cytokines for that reason. So while TNF blockage is quite useful in certain diseases, especially if TNF production is induced for a longer term, in a septic reaction, there is no use of blocking TNF because by the time clinical signs appear, usually the TNF alpha is already gone. So it cannot be used neither for diagnostic purposes nor for therapy, the blockage of TNF in sepsis, but it can be used in chronic inflammatory diseases when there is a constant TNF production. So Kevin Tracy afterwards decided that it's another kind of material which must be present in our body, which is responsible for the lethal action if TNF is gone by a couple of hours. So what is responsible for the later death of animals if this TNF is gone? So he decided to look for changes in the plasma, and that's how he found HMGB, a protein which we now know act as a damp. This one is produced much later than TNF, and there will be a steady elevation in the serum level of HMGB for hours, for days. So he tried whether HMGB could kill the mice the same way as TNF. The only discrimination we made is that he still used a little bit of LPS and added huge amounts of HMGB to that one, and that could kill the mice, though this very tiny amount of LPS wouldn't kill the mice by itself. And when he used the anti-HMGB antibody with the tiny amount of LPS, that one could protect the mice just as well. So the conclusion was that HMGB was also involved in this inflammatory reaction, but unlike TNF, it's a later mediator of the reaction. So HMGB acts as a damp just as well, not only as a cytokine, which will carry the message. When the cells are treated with TNF, macrophages will produce TNF in response to HMGB. Interestingly, when he used various knockout mice, it turned out that TLR4, the very same TLR4 was the receptor of HMGB as that of LPS. It also acts on RAGE receptors tiny bit, but not as much as on the TLR4. So if an MD2 knockout mouse is treated with HMGB, there is no TNF production, only in the wild-type mice. So from this one, it's quite clear that actually it uses not only the same receptor, but it's the same co-receptor. And later on, it turned out that HMGB binds to the MD2 portion just as well as LPS to induce the reaction. Not all forms of HMGB is active, so a redox-regulated protein is needed for the activation. If there is a very strong oxidative stress, that one can reduce the activity of HMGB, whereas if HMGB is just released from the cells, this one acts as a normal transcription factor normally within the cells. There is nothing because only the redox-regulated disulfide form is active. So the fully reduced or the fully oxidized molecule is inactive. So it is a prototypical DAM, but it also acts as a late cytokine. So in general, we can say that the danger signals include, for example, LPS from infections or heat shock proteins, HMGB from tissue damage. This one will act on specific receptors, for example, on the TLR4 on macrophages, which are the sensor cells of the danger signal. Then they will produce inflammatory cytokines like TNF and IL-1 and IL-6, and also HMGB, and HMGB will act on the same cell and will act as an amplification cycle if there is a huge amount of activation. And this one will induce the vascular reaction and the effector cell mobilization and will attract the cells to the side from the bone marrow, and they will remove the injured cells and kill the microorganisms. Now let's go a little bit more into details because we talked about the gram-negative bacteria only. Gram-negative bacteria will have the lipopolysaccharide, but we do know that there are many gram-positive bacteria which are also pathogenic. So what's the difference between the two of them? In the gram-positive bacteria, there is no lipopolysaccharide, but they do have a lipoteichoic acid, which goes through the cell wall. They have cell walls, and that's the reason why they can be stained by the gram stain, that one will retain the gram stain, whereas the gram-negative bacteria will not have a cell wall, so they do not retain the stain. So there is a different kind of protein molecule present in the membrane of the cells. Our body uses the TLR4 to detect the gram-negative bacteria using the LPS as a signal. Gram-positive bacteria, on the other hand, are detected via the lipoteichoic acid, via the TLR2 receptor. TLR2 is somewhat similar to TLR4, and this one will also induce imidazole activation and NF-kappa B activation. So by the time we can see the inflammatory reaction, we cannot really tell whether this one is caused by a gram-negative or gram-positive bacteria because there is no difference. And it will also induce the inflammation in the very same way. So the only difference between the two of them is what kind of receptors the body uses. So we have specific receptors for various pathogens. Actually, we have quite a few other receptors in our cells, and collectively we call them pattern recognition receptors. These pattern recognition receptors are the major receptors of the innate immune system. They are present on the cell membrane, for example, the TLR molecules, or the lectin receptor, is on the cell membrane. Bacterial components are detected mostly by TLRs on the cell membrane, and fungal polysaccharide by the lactate receptor. But we also have intracellular receptors, like the node-like receptor family and the ring-like receptor, and also endosomal intracellular TLR receptors are present in our body. So these receptors are used to detect any damp or PAMP in our tissue. No distinction is done afterwards. They will look almost the same. The inflammatory reaction will be very similar. Let's see the toll-like family, the largest family and the most known currently. TLR family includes cell surface receptors, for example, the TLR2 and TLR4, which will detect the LPS and the lipoteichoic acid. But it also includes the TLR1, TLR2 complex, which will detect triacylated lipoproteins, the TLR5, which will detect flagellin, and the TLR2, TLR6 heterocomplex, so which will detect the diacylated lipoproteins. And they will all signal through the midosome, and they will all activate the inflammasome the very same way. Endosomal reactions are very similar to this one. Endosomal TLRs include TLR3, TLR7, 8, and 9. The TLR7 and 8 will detect single-strand RNA molecules. The CpG DNA will be detected by the TLR9, and that will induce the midosome and the NF-kappa B signal the very same way as the cell surface receptors. The only exception is the TLR3. TLR3 will detect double-stranded RNA molecules, and that's the antiviral detection system. That one will detect double-stranded RNA molecules and will induce IRF3 translocation and interferon production, type 1 interferon production in the nucleus. And this is the so-called antiviral response, which is slightly different from the inflammatory reaction, but all viral infections will induce this interferon response. So if we want to make just a comparison of how these TLRs work, we can say that there are TLR receptors on the cell surface. They will detect various exogenous molecules, mostly derived from bacteria, either gram-positive or negative bacteria, but some of them will also detect a few viral envelopes, fungi, or protozoa. Similarly, they will also sense the tissue damage by detecting heat shock proteins, HMGB. The intracellular TLRs, the endosomal ones, will mostly detect double-stranded, single-stranded RNA and CpG DNA, will mostly detect the viral infection, but will be also activated with bacteria, fungi, protozoa as well. And they will detect cell damage whenever there is a mitochondrial DNA released from the mitochondria within cells, or single-stranded RNAs will appear within the cells. The question arose afterwards that how can so few receptors detect everything? So if you compare it to the adaptive immune system, the innate immune system works with very few receptors. Only a handful of them is present, which can detect so many signals. So how does it work? Well, the receptors definitely are imperfect. So they will not detect all the signals, all bacteria, with the same efficiency. Some of them are detected much better than the others, as represented on this figure. So while some of them are very well detectable, others may not be detected at all or may not be detected as efficiently. So how do we know which ones are to be detected and which ones are not to be detected, because we also have a microbiome in our body. Well, the trick is that this one is only the structural detection system of the innate immune system, and it's not the only way. We believe that there is a homeostasis associated molecular processes receptor in our body, which will sense the functional changes, which can be there whenever there is an infection or tissue injury. It means that while the pathogens or the tissue injuries will release DAMs or PAMs, and they will be sensed by the pattern recognition receptors, we also have HAMs. For example, the ATP level will drop in the cells, the potassium level will change. These HAM sensors will detect these changes, and whenever there is a secondary signal coming from the pattern recognition receptor, that is the one which will induce inflammation. If this one is missing, or that part, there will not be a truly important inflammatory reaction. So to see one of the examples of HAM sensing, here is the pyrine, which is more or less an inactive protein in the normal cells. There's a guarding protein, the 1433, which is constantly in a phosphorylated state and keeps the pyrine inactive. This one involves a phosphorylation cascade. This kinase cascade will keep it phosphorylated. But what happens if the cells are ATP depleted? They will not act on the guard protein, and the active pyrine will be instantly released, and an inflammasome will be assembled. So this is like we were on a minefield where we have a bomb which can be very rapidly turned on, and whenever this the danger signal triggers this bomb, this one will explode. So that's how the inflammasome can be activated so rapidly, and that's how the HAMP-mediated signal comes in hand in hand with the pattern recognition signal. So what are the cells which will detect these signals? First of all, we can classify the various cells of the innate immune system into sensory cells, sensor cells or sentinel cells, effector cells, which will kill the germs, and regulator cells, which control the whole immune response. From the sensor cells, the most important ones are the tissue-resident macrophages. To a certain extent, dendritic cells are also involved in this process, and they will help the tissue-resident macrophages. And the mast cells can also help a tiny bit, but they are less important. Effector cells include neutrophils, monocytes, and the NK cells. And regulatory cells are the so-called innate lymphoid cells. And next to that, we have the basophilic cells and the eosinophilic cells, which are also act as regulators of the response, but also act as effectors. So let's see the macrophages. Actually, we have macrophages all over our body, next to the endothelial cells around each of the vessels. Some of them also protrude into the circulatory side. So for example, in the pancreas or in the liver, the Kupffer cells will go inside the lumen of the vessels. These are naturally the first cells to encounter any damage because they will sense it immediately. And since the micro vessels are tiny, they are just a single layer of endothelial cells, and they are in close contact to almost all of our cells, it's evident that these macrophages are everywhere in our body and can detect everything immediately. So these are the first ones to see the signals. Some of them have specific names. For example, in the liver, we call them Kupfer cells, in the skin, Langerhans cells, or the microglia cells in the central nervous system, or osteoclasts in the bone. Others are simply called adipose tissue, macrophages, and so on. These are the cells which are always there. They will sense the danger signals, and that's how they will be turned on. They will be activated and start up the inflammatory reaction. There is also a secondary alternate activation process, which is known, which is involved in the activation of the wound healing, the later stage of the inflammatory process. This one will happen in response to certain interleukins, interleukin 4 and interleukin 13. The important part is that these cells are not coming from the bone marrow. Actually, they originate from embryonic precursors, and they establish a tissue-specific pool of these macrophages within each of our tissues, and they locally renew themselves. So whenever there is an irradiation, a chemotherapy which will be locally delivered, that one will kill the local macrophages as well, so inflammatory reaction will not be that harsh, but it means that the protection will be gone as well. Certainly, they can be reestablished once again from the bone marrow, but it takes time. Dendritic cells, on the other hand, will just help the macrophages, and they are derived from the bone marrow origin. Neutrophils. Neutrophils are the most important effector cells. They make up approximately 70% of the leukocytes in the blood. They are very short-lived cells. They only live for 7 hours, no longer in the circulation, but it takes a much longer process to generate them. Neutrophils disappear in 3 places in our body: in the spleen, in the liver, and in the bone marrow. Since the number of neutrophils can change markedly in response to danger signals, we can use them as an excellent marker of infection or inflammation, and that's the major use of them. They will kill the microbes via secretion of their granules. These granules of the cells contain various enzymes and toxic materials which can kill the germs, or they can phagocytose the microbes and can lyse them within the cells. They can produce reactive oxygen species, and those combined can also kill germs. And they have a fourth way to kill the germs, and that's the netosis, which is responsible for the extracellular killing mostly. Neutrophils can be turned off by 2 steps. So even though when they circulate in our body are mostly in the basal quiescent state, they are not activated immediately. They do need 2 steps to have full activation. It's like a safety switch. So they need a first activation, priming, and they will need a secondary activation, which will turn the activity of them to full activity. So where are they coming from? From the bone marrow. They are generated from hematopoietic stem cells through a long maturation. And this one will include cells you will learn during the second term. The mitotic pool in the bone marrow is making up approximately 10% of the cells, whereas we have the majority of all our neutrophils in the so-called storage pool. Approximately 90% of all of the neutrophils of our body is in the storage pool at any moment. And that one is the explanation why the number of neutrophils can be increased so rapidly to such a high level, because these neutrophils can be pushed into the circulation whenever they are needed. Within the circulation, where they normally have only a 7-hour half-life, they are in the circulating pool, or some of them in the marginating pool. And when there is an inflammation, there is one way to push them into the circulation from the storage pool, from the bone marrow. And this one can induce a large increase in the neutrophil count. So 11,000 white blood cells might be present in the blood, or even higher number. Also, the marginating pool can be pushed into the circulating pool, so the number of the cells can be increased, and the survival of the cells can be prolonged to up to 1 full day. So all of these are responsible for the huge elevation of neutrophil count in the blood. We also call it a left shift when these earlier types of cells, which do not have the 4 or 3 to 5 lobules of the nucleus, will appear in the circulation in larger quantities, because we normally throw them to the left. So that's why it's called a left shift. And in certain cases, the inflammatory reaction is such an excessively active process that the leukocyte count can go up above 25,000, and that's the time when we call it a leukemoid reaction. So neutrophils will have primarily granules with myeloperoxidase, and they can phagocytose the bacteria, just like here on this scan. The bacteria are on top of the neutrophil and they are internalized. In the phagosome, they can lyse the bacteria and they can produce oxidants to inactivate them by the NADPH oxidase. This one will produce superoxide, which can be turned to hydrogen peroxide, which has much longer half-life, and can be combined by chloride by the myeloperoxidase, and then hypochloric acid can be produced, which has even more toxic effect and longer half-life. When released into the environment of the cells, this one can kill bacteria in the close neighborhood, but only in close proximity. So this one is not responsible for the killing of germs in large quantities in the extracellular space. NETosis is responsible for that one. So here you can see this gnat produced by the neutrophils and the bacteria inside the gnat. So how is it produced? The cells have this very specific segmented nucleus, which means that these lobes of the nucleus can be pushed out of the cells as an active process, and then the whole DNA come out, as you can see here on this one, and this one will also include the granule material, so the enzymes and toxic materials will be in this whole gnat formed by the DNA, and the bacteria will just attach to that one and by the enzymes will be killed. So that's the way how neutrophils can get rid of extracellular microorganisms. Monocytes, on the other hand, are slightly larger cells, and we have much fewer of them in the blood. Only 4 to 10% is the normal proportion of monocytes. They have a longer half-life, and we discriminate 3 kinds of phenotypes of monocytes, but the great majority of them are classical monocytes, which can detect with CD14 the LPS. They are important in the phagocytosis and cell killing, and since they are larger, they can internalize even larger tissue damages or external bacteria. These are the cells which will turn up much later as macrophages in the tissues because they can differentiate it into inflammatory macrophages. But these are not responsible for the origin of the tissue-specific tissue macrophages. Now, macrophages can also induce an antiviral response, which is slightly different from the inflammatory reaction. Naturally, viral envelope can be detected, for example, by cell surface TLR molecules, and that one can induce an inflammatory reaction. Or they can be internalized and can be detected by the intracellular endosomal receptors within the cells, and that one can induce the same inflammatory reaction. TLR3 can detect the double-stranded RNA, and that one is responsible for the true antiviral response, the interferon response induced by cells. Also, the inflammasome activation is almost the same in this case. So any damage which is induced by the viruses will result in inflammasome activation. So macrophages will induce an antiviral response and will induce interferon production. This interferon production will change our structure of cells. That one will stimulate the production also of interferon gamma and will induce very important expression changes in the cells. Endothelial cells, fibrocytes, epithelial cells will all change the gene expression. This one will happen through open chromatin regions, which will be present in this case. So many genes can be accessed at the same time by this method. So when the macrophage is infected, detects the infection, that one will induce a protective response in the fibrocytes, endothelial cells, and epithelial cells. This one will include 3 important steps. MX proteins are expressed in the cells, and these proteins will form a scaffold around the viruses, which will block the viruses and the viral replication within the cells. They will also produce RNases, and this one will cleave the viral RNA and will also activate the translation initiation factor blockage. So this one will inactivate the translation of proteins, viral proteins, and the production of new viruses. Now, the true effector cell against viral responses is the NK cells. NK cells make up 10% of lymphocytes in the blood, and they have a half-life of a couple of weeks. They are not true lymphocytes, but rather lymphoid cells because they do not express T or B cell receptors, but they have the Fc receptor on the surface. They recognize infected cells that downregulate MHC1 molecules. That's a common response to viral infections, that the MHC1 molecules are downregulated, whereas interferons will stimulate the MHC1 expression. So in healthy cells, NK cells will see the MHC1 present on the surface. And this one will block the action of NK cells as an inhibitory signal, but they will also have activating receptors on the surface, which are not present in healthy cells. These are virus-induced proteins or certain proteins which might be present in stressed cells. So in a healthy cell, the MHC1 signal, the presence of inhibitory receptor, and the lack of activating receptor will block the NK cell activation. On the other hand, in infected stress cells, where we have multiple virions, and the MHC is downregulated, the NK cells will not detect the inhibitory receptor, the MHC1, but they will detect the activating receptor, with activating receptors, the activation signal, and that one will induce degranulation of the NK cells, and they will kill these cells. So by recognizing the missing self. Innate lymphoid cells are the two controllers of the inflammation, and we'll go through this one very rapidly. We still don't know too much about the innate lymphoid cells. Currently, we classify them into three major groups. They are lineage marker negative cells, also come from the bone marrow, but they are different than B cells and T cells. They have a common lymphoid precursor just as well, but they develop slightly differently. And the three distinct populations, what we discriminate right now, are discriminated based on their transcription profiles and cytokine production. They are termed ILC1, ILC2, and ILC3. ILC1 cells also include the NK cells. So these cells are the ones which will be responsible for the immunity against viruses, but they also work against certain bacteria and parasites. ILC2 cells mostly work against helminths and allergy. They are involved in that one. They will produce IL-4, whereas the ILC1 will produce TNF and interferon gamma. ILC3 cells mostly produce interleukin-17 and IL-22. They work against extracellular bacteria mostly, and we still don't know how they can regulate all the responses, but if there's a disruption of the processes, we can have a false reaction and we can have an inflammatory reaction maintained for a longer period of time in our body. So altogether, the whole inflammatory process is rather simple. We have macrophages or other sensor cells which will detect, using the danger signals, the various insults. So either viral infections or bacterial infection and so on will induce cytokine production, DAMP release, and also HAMPs in our body. And these will act on the cells and will amplify the signal. Altogether, this one will mobilize the neutrophils or monocytes, the complement system, the innate lymphoid cells, and will induce fever and vascular changes. And the microbes are removed from our body as a response to that one. And normally we can see a resolution or wound healing. If it doesn't work, the adaptive immune system will be turned on with help of the innate lymphoid cells. So we can have sensors, we have messengers, and effectors in our body. So how can we apply this one to the clinical practice? First, the diagnostic use. Let's see. We've seen that certain clinical signs can be easily detected, like fever, pain, and redness, or response to the adequate treatment. But we also have specific lab results which can be used. For example, the leukocytosis, neutrophilia, can be very simply used because the neutrophil count will be elevated. We also see a raised SED rate in infections and inflammation and raised CRP. And sometimes we can use a blood culture or a positive serology test. So what are these? The SED rate and the CRP. In response to the cytokines, the liver will change the expression of multiple proteins. The liver is responsible for the majority of the proteins in our plasma. Some of the proteins are downregulated. We call them a negative acute phase reaction, as negative acute phase proteins, these group. Others are upregulated, so we call this change a positive change, positive acute phase proteins. As a consequence, mostly the globular proteins will increase and albumin will be decreased. As a result, the sedimentation rate will change. Globulins will attach to the surface of red blood cells, and the red blood cells fall much quicker in the tubes. So what are these negative and positive acute phase proteins? The most important ones are albumin, transferrin, antithrombin in the negative group, and the C-reactive protein, hepcidin, ferritin, fibrinogen, prothrombin, factor VIII, von Willebrand factor, and plasminogen activator inhibitor 1 in the positive acute phase group. We'll discuss the role of this one, the transferrin and ferritin in iron deficiency anemia, and the hepcidin in the anemia of chronic disease during the second term. The bottom ones are involved in the procoagulant state, which is always present in inflammatory reactions. So what is the SAD rate? It's a very old-fashioned test, but it's still very useful because it needs nothing more than some blood and an anticoagulant, and then a specific tube, the Westergren tube. So the red blood cells will normally fall slowly down in a tube, but not too rapidly because they have an external charge which will block it. But once you have various proteins which will attach to the surface of the red cells, that one will induce rouleau formation of red cells, and these rouleaus will fall down much quicker. So very simply, this one is measured in millimeters. How much is the fall of red cells within that time? Not inflammation is the only process which will increase the sedimentation rate. Naturally, pregnancy, cancer, anemia, and also myeloma will increase it very much. And it's also dependent on the gender and the age a tiny bit, but actually it's a very simple test, very cheap, and still can be used as an additional method in our diagnostic repertoire. So in inflammation, the SAD rate is raised due to the increase in the globular proteins. CRP. CRP is a good marker of infection or inflammation because this one is upregulated in response to cytokines, mostly through IL-6 in the liver. This one is a protein which was derived from the C fraction of proteins induced mostly by the pneumococcus originally. So that's why it's C-reactive protein. It acts as a pentoxin, and we do use it as a diagnostic test. It's not that important for its function. So in response to the cytokine response, CRP is produced. This one will bind to proteins and will induce complement activation, and by that one, the complement will bind to this complex and will induce opsonization, or will induce the assembly of the membrane attack complex, and the bacteria will be lysed. The half-life of CRP is a little bit longer, so it's not worth measuring it every day. It's rather measured every second day. So there is one more thing, which is not truly a lab result, but rather a clinical sign, but it's not explained fully by the changes so far. It's not because of hyperemia. It's because of a reaction in response to cytokines. So in response to the cytokines produced in the brain, in the hypothalamus, prostaglandin E2 will be upregulated, and that one will induce, which might be blocked by antipyretics and glucocorticosteroids, this one will induce thermogenesis, a non-shivering thermogenesis, which will result in fever. So how will it induce fever? Naturally, almost the same way as our normal heat production works. We do use oxidative stress in this case to activate the uncoupling proteins in the muscle cells. And by using up the mitochondrial potential for thermogenesis will not produce that much ATP, but will dissipate heat. We use UCP3 because that's present in the muscle cells. And through UCPs, a proton leak will be produced. So the protons will be released from the intermembrane space back into the matrix without ATP production. So heat will be generated from the stored energy. This one will destroy the mitochondrial membrane potential. So altogether, if someone has high fever, he will feel weak and tired. So UCP1 is the one which is responsible for the normal thermal regulation, for the normal heat production of our body. UCP2 is mostly involved in the redox regulation, and UCP3 in the skeletal muscle is responsible for the fever. Fever production is not produced in mice which do not have the UCP3. In UCP3 knockout mice, in response to LPS, you don't see the elevation of heat because this is responsible for the heat dissipation. So it means that the cytokines released into the circulation will induce prostaglandin E2 production. That one will also upregulate UCP3, but will induce an oxidative stress and UCP3 activation, and this one will activate the mitochondrial proton leak via UCP3 and will result in heat production. There are the best antipyretics, non-steroidal anti-inflammatory drugs, and also glucocorticoids will reduce the fever. There are two diseases you've got to know: Addison's disease. In Addison's disease, a so-called Addisonian crisis may occur, which will be associated with high temperature, very high fever, because actually the patient will not have glucocorticoids in the body, and this one will not block the prostaglandin E production. The other one is the thyrotoxic crisis or thyroid storm and hypothyroidism. Thyroxine overproduction will upregulate UCP3 and the patients will not tolerate the heat. But this one can also induce very high heat production in the thyroid storm. And since we are below our usual step where we normally interfere with heat production, it's very difficult to interfere with this one. But it also means that the old-fashioned vitamin C usage, antioxidant usage, is quite effective to have some additional effect in most of the cases. So in a simple infection, we still recommend taking some vitamins, some extra vitamins compared to the usual supplementation. And we'll return to this one. Glucocorticoids, steroids, and non-steroidal inflammatory drugs not only block this step, they may act at an earlier step in inflammation. So how can we interfere with this one? Inflammatory reactions. The majority of our drugs are claimed to be COX inhibitors or phospholipase A2 inhibitors. Antibiotics act against bacteria or antifungal drugs and block the fungal infection. Antiprotozoal drugs will kill the protozoans, or antiviral drugs may act against viruses. Chemotherapy will reduce the tumor cell lysis or will the reaction to these. Immunomodulatory drugs may block the cytokine response, or if we can use anti-cytokine drugs, we can block the action of cytokines. Chemokine and selectin inhibitors would block the neutrophils theoretically, but they are not used because they are more or less useless. And we do hope that in the future we'll have anti-TLR antibodies, which will block the early reaction of inflammation. And we'll probably have HMGB1 inhibitors in the future. We still don't know whether we'll have inflammasome blockers or not. Currently, we usually have a stepwise treatment. So non-steroidal drugs are the first ones we use: simple aspirin, paracetamol, Tylenol, or any other COX inhibitor can be used. If this one is not enough and they are off-shelf available, we may use a glucocorticoid steroid. They require a prescription all over the world. They are stronger anti-inflammatory drugs and they will have wide-ranging side effects which are not preferred. If we cannot really block the inflammatory reaction with steroids, we can use a third step. We can use immunomodulatory drugs, anti-cytokine therapy, anti-TNF alpha treatment, for example. So what else can we have? We do have TLR blockers, anti-TLR2 and TLR4 antibodies in trial phases. Hopefully they will be in practice rather soon and they will work against many things. We also have HMGB inhibitors under development, but actually they might not be better than the currently available drugs, which will also have some HMGB blocking activity. We do not have midosome inhibitors right now or NF-κB inhibitors, and we do have anti-cytokine drugs, but this one mostly includes the anti-TNF antibody infliximab. Anti-inflammasome drugs or inflammasome inhibitors are under development, but we still don't know what will happen. So the strange thing is that over recent years, it turned out that many of the COX inhibitors or non-steroidal anti-inflammatory drugs or some other agents, what we commonly use, for example, metformin, will also have an anti-HMGB activity, or they can block HMGB very simply. So aspirin is not only blocking the COX enzyme and the prostaglandin production, but will also act by blocking the early step of inflammation by blocking HMGB partially. Also, metformin and even heparin can do the same thing. On the other hand, HMGB1 might be indirectly blocked by certain agents, for example, glucocorticoid steroids. Glucocorticoid steroids block the reaction to HMGB within the macrophages at the very early steps. So these will also work against HMGB and the early step of the inflammatory process. The even stranger thing is that simple antioxidant drugs like vitamin C and N-acetylcysteine, which are commonly used, can inactivate HMGB because the fully reduced form of HMGB is just as much inactive. Conservative treatment options are also present. For example, necrosectomy or abscess drainage can be used. So the drainage or even limb amputation might be an option in certain cases. They are not the opposite of antibiotic treatment, though quite often they are believed to be just the alternative option to conservative treatment. They do remove the damaged tissues. They protect against the excessive damp and sometimes PAMP release into the circulation. So that is the main reason why we do need them, and we will need them in the future as well, because we've got to get rid of the danger signals. So let's see finally 3 very simple examples everyday examples. Common cold, which is known to everyone because it happens to everyone a couple of times each year, commonly in the winter months mostly. We do know that this one will result in a blocked or runny nose, a sore throat, and we may experience pain on swallowing, and we may have voice breaks and feel malaise. Subfebrillity or even fever might be present, and we will cough, or sneezing will be the presenting symbol. The cervical lymph nodes will be tender, and quite often muscle pain or joint pain will be present in response to viral infections. The etiology is mostly viral by airborne viruses. Rhinoviruses, parainfluenza virus, adenoviruses, or certain coronaviruses, not the SARS-CoV-2, but the other ones. They will induce these symptoms, and very, very rarely, this might be induced by group A streptococci bacteria. So what shall we do in case of a common cold? Well, naturally, most of the time, there is no medical help needed in this case, but if someone presents with these symptoms, a simple history taking is needed regarding the presenting complaints, the history of the presenting complaints, past medical history, what kind of drugs the patient takes, and a physical examination which can be limited to the head region. No blood test is needed normally, if you don't need to worry about a coronavirus pandemic. And no specific treatment is needed. We just use the so-called symptomatic treatment. We do recommend to take some rest because everyone will be tired if the virus will use the energy in the muscle. Or take some vitamins to block some of the reaction. Use a nasal drop, a strepsil, an antipyretic, for example, Tylenol or aspirin or any of them, and if needed, an N-acetylcysteine can be used. Bacterial origin is the only suspicion which occurs if the symptoms will last much longer or there is excessive pus formation can be seen. In that case, amoxicillin is the preferred drug. So let's see the other one, an acute diarrhea. In an acute diarrhea, which is again a very common disease, and an average adult is estimated to have 1 or 4 occasions each year. We call it acute diarrhea if it will not last longer than 14 days, but by definition, but most of the time it will not last more than 1 or 2 days. The symptoms are clear. Loose stools might be present with some subfebrillity, some mild abdominal pain, and occasionally nausea and vomiting may occur. Etiology is mostly infectious by fecal-oral transmission and mostly by viruses, rotaviruses, noroviruses, adenoviruses. Much less commonly, it might be induced by bacterial or bacterial toxins like Shigella, Salmonella, Campylobacter strains, or certain E. coli strains. Occasionally, it's also occurring as a reaction to stress or as a response to drugs. So what do we need to do if medical help is needed? First of all, we just need to rapidly assess the presenting symptoms, whether there is any exposure to certain foods, travel, which may change the picture, and we've got to know what kind of drugs the patient takes. The most important part regarding the physical examination to estimate the water loss and to suspect whether there is any more severe abdominal disease present or not. No blood tests are necessary most of the time, and the symptomatic treatment can be used, which means fluid replacement, mostly oral rehydration, and a simple diet can be used. If the symptoms cannot be blocked any other ways, then we can use Imodium, for example, and antibiotics are very rarely needed. Only in the immunocompromised host it's recommended. Otherwise, this one will have a much worse effect, and we will try to avoid them. In children, probiotics are generally recommended. So again, it's a viral infection. We don't need to worry. We do know that in our antiviral response, the innate immune system will take care of the disease and the symptoms will be gone. This one will also induce the activation of the adaptive immune system, which will prevent the reinfection of the individuals. Now let's see the third example, the cystitis. This urinary tract infection is the most common cause in the general practice that requires antibiotic treatment. It's quite prevalent in females. It's estimated to be around 11% in the whole population. And the symptoms are well known: dysuria, a painful micturition with burning sensation or discomfort sensation is quite common, with frequency and urgency. Additionally, it may induce some mild pain or tenderness, and rarely it will result in hematuria. The etiology is almost always bacterial. An ascending infection, bacteria from the intestinal tract go in a retrograde fashion up into the bladder. It's much more common in females naturally than in males. In the great majority of the cases, it's caused by E. coli. To a lesser extent, it might be caused by Enterococcus or Proteus or Klebsiella strains. But since it's mostly caused by E. coli, that's the major bacteria what we've got to treat. So what do we need to do? A very simple history taking is needed, because the presenting symptoms will help us immediately to discover this disease. We do need to know the past medical history and drug history to make sure that we are not reusing just the same drug, which already led to the infection. The gold standard method is the urine culture, but it's not necessary all the time. Based on the symptoms, we can decide whether treatment is necessary or not. So a simple dipstick, which will detect the nitrite level, can usually detect the presence of bacteria. So the treatment. First of all, we do need to recommend high fluid intake. That one will wash the urinary tract, and we will use antibiotic treatment because this is an infection where the innate immune system cannot really get rid of everything. So we do need an antibiotic treatment to assist our immune system. We use fosfomycin or ciprofloxacin. In the elderly, even nitrofurantoin might be used, and trimethoprim might be used, especially in kids. And that's it. So the innate immune system and inflammation are very important. Almost all the diseases will have some involvement of the innate immune system, because this is the first line of defense of our body. The reaction is very rapid and very effective, because we can get rid of the majority of the bacteria, or the majority of the tissue damages without any further help. But it also means that if there is a disturbance of the innate immune system, that one will lead to infections or even autoimmune diseases. And if the system is overly activated, this one will result in a septic reaction. Thank you for your patience.